Today, I'll be discussing my graduate research focused on assessing nitrogen critical loads at North Cascades National Park Service Complex. So, transportation, industry, and agriculture all emit reactive nitrogen into the atmosphere. Nitrogen emissions from transportation and industry are dominated by nitrogen oxides, while emissions from agriculture are mainly reduced reactive nitrogen forms, namely ammonia and ammonium. Nitrogen emissions can be transported and deposited back onto terrestrial and aquatic ecosystems as atmospheric nitrogen deposition. Over time, excess nitrogen deposition alters biodiversity and plant and soil chemistry with cascading effects through ecosystems. Anthropogenic nitrogen deposition has already contributed to ecosystem changes across the Western US, including disruptions in nutrient cycling, ecosystem functioning, and shifts in community composition. So even minimal increases in nitrogen deposition can potentially impact sensitive nitrogen-limited ecosystems. Nitrogen critical loads can be developed to assess the threat of nitrogen deposition to ecosystems. A critical load exceedance occurs if ambient deposition is greater than a defined critical load, and it implies ecological changes specified by the critical load would be anticipated. Identifying areas where current deposition exceeds critical loads can help air quality regulators and land managers determine where emission reductions are most needed. Accurate measurements of nitrogen deposition are needed for land managers to determine nitrogen critical loads and exceedance estimates. The deposition estimate used to evaluate a critical load can influence whether an exceedance is expected and can change management and policy response. Typically, nitrogen deposition estimates come from ambient monitoring, atmospheric modeling such as CMAC, which Adrian talked about yesterday, and a combination of both known as a model data fusion approach. In addition to ambient monitoring and atmospheric modeling, biomonitors such as epiphytic lichens can be used to assess and predict nitrogen deposition. Lichens have long been useful indicators of air quality because they have a high surface area and lack roots. They readily absorb water and lack specialized barriers to prevent vapor loss. And there is a range of air pollution sensitivities across species. Epiphytic lichens, meaning lichens that grow on shrubs and trees, obtain their nutrients from air, precipitation, and substrate runoff. This means that tissue levels of nitrogen are correlated with ambient atmospheric levels of nitrogen. The nitrogen concentration of epiphytic lichens can be utilized to monitor nitrogen deposition in forested areas as a low-cost method to get accurate measurements of nitrogen deposition across a fine or broad spatial scale, even one with complex topography. A study by Root et al. 2013 co-located through-fall deposition collectors with collected lichen tissue samples at 84 study sites across Western North America. And here you could see a regression that they developed between through-fall nitrogen measures and nitrogen concentration in Platysmacia glauca, which is one of the lichen species collected in their study. The concentration of nitrogen in Platysmacia glauca is along the x-axis, and dissolved inorganic nitrogen in kilograms per hectare per year is along the y-axis. This regression model effectively estimates through-fall inorganic nitrogen deposition from lichen nitrogen concentration. And then these nitrogen deposition estimates can then be used to identify areas that are in exceedance of nitrogen critical loads. So, why is my research necessary? As I discussed earlier, accurate measurements of nitrogen deposition are needed for land managers to determine nitrogen critical loads and critical load exceedances. However, nitrogen deposition estimates from atmospheric models, such as CMAC, can have a high degree of uncertainty, especially in complex terrain. The model data fusion approach is still reliant on measured values that come from instrumented monitoring sites that are sparsely located throughout the Western US. So there's a need for greater accuracy in nitrogen deposition estimates, especially across the Western US, where there's a vast array of public lands that may contain ecosystems that are sensitive to elevated nitrogen deposition. Based on these gaps in knowledge, My research is focused on increasing our understanding of patterns of atmospheric nitrogen deposition in the North Cascades, determining the extent to which ecosystems in the park are being affected by nitrogen deposition, and gaining insights into predominant sources of nitrogen emissions affecting the park. To orient you with my study area, North Cascades National Park Service Complex, it's in the northwestern corner of Washington, just below the Canadian border. And it's a park known for its jagged peaks crowned by more than 300 glaciers. It spans the Cascade Crest from the temperate rainforest of the wet west side to the dry ponderosa pine ecosystem of the east. And it consists of several units, including the north and south units of North Cascades National Park, as well as Ross Lake and Lake Chelan National Recreation Area. I established lichen air quality plots across the North Cascades. I identified 10 distinct spatial areas across 3 transects that represent both north and south and west to east gradients within the park. At each one of these spatial areas, I established 3 plots along an elevational gradient. The lichen air quality plots were 114-foot radius circular plots with an area of 0.9 acres. And at each one of these plots, I collected lichen tissue samples for elemental analysis according to U.S. Forest Service protocols. Here are a few photos of me collecting lichen tissue samples in the North Cascades. And afterwards, I brought these samples back to the WSU Stable Isotope Core facility where I worked on processing them and analyzing them for both nitrogen content and nitrogen stable isotope composition. For an initial interpretation of my lichen nitrogen content from the collected samples, I compared my values with the provisional element analysis thresholds for percent nitrogen for my focal species, which was determined by the U.S. Forest Service. Values above these concentrations are considered elevated, and values at or below these concentrations are considered within background range. And here you could see the provisional nitrogen threshold for Platysmacia glauca, and that's 0.59, and a nitrogen content above this value would indicate elevated nitrogen deposition, and a value below would indicate background levels of nitrogen deposition. This figure is showing lichen nitrogen percent data for 2 of my focal lichen species, Platysmacia glauca in gray and Alectoria sarmentosa in green. Lichen nitrogen percent is along the y-axis, and the longitude of the air quality plots in decimal degrees is along the x-axis, going from west to east across the park. And I added these dashed lines to show the value of the provisional thresholds for both species, 0.59% nitrogen for Platysmacia glauca and 0.65 for Alectoria sarmentosa. We see that all the points fall below those respective thresholds, indicating that these plot locations likely do have low background levels of nitrogen deposition. However, we do see some points with values upwards towards these thresholds, indicating that these areas may have slightly higher nitrogen deposition. So looking at this data spatially, just focusing on one of the collected lichen species, Platysmacia glauca, we can see which areas of the park have the highest percent nitrogen values, suggesting higher rates of nitrogen deposition. The color ramp here goes from dark blue to red, going from low to high percent nitrogen values. And we could see this area here in the southwestern region of the park, which is the Cascades Pass area, has some of the highest percent nitrogen values for Platysmacia glauca, upwards to 0.5% nitrogen, which is just slightly below the provisional threshold for this species. So I'm now going to show that same map displaying percent nitrogen values for Platysmacia glauca overlain over the National Land Cover Database layer, zoomed out to show the region surrounding the North Cascades. So here are the land cover classifications. We see that the areas immediately surrounding the North Cascades are mostly forested, but in close proximity, there's acreage that is light yellow, indicating pasture for hay and livestock, dark brown, indicating cultivated crops, and varying shades of red, with light red being low intensity development and deep red being high intensity development. So the western edge of the park is in closer proximity to these human impacted land cover classifications. This includes the Cascade Pass area that I pointed out here that had the highest percent nitrogen values, and it's situated closest to this large red shaded area here, which is the Seattle metropolitan area. So proximity to nitrogen pollution sources, as well as landscape features and meteorological variables, are likely influencing rates of nitrogen deposition in the North Cascades. Analyzing lichen nitrogen content will help us assess patterns of deposition across the North Cascades. I'll also be assessing the nitrogen stable isotope composition of collected lichen samples to be able to assess dominant sources of nitrogen pollution. Just a real quick primer on stable isotopes. Nitrogen has two stable isotopes, 14N and 15N, which differ in their number of neutrons. And you can see here that the lighter isotope, which is 14N, is much more abundant than the heavier isotope, 15N. And the notation for nitrogen stable isotope composition is delta 15N, which is expressed in units of per mil. And essentially, we're looking at a ratio of ratios. So R is the ratio of the heavier to light isotope, or 15N over 14N, and then the nitrogen stable isotope composition of a sample is the R of the sample over the R of a standard, which for nitrogen is air, which is 0 per mil, minus 1 multiplied by 1,000. So isotopes of reactive nitrogen species can aid in understanding emission sources and chemical transformations that contribute to reactive nitrogen deposition. And this figure is from a recent review by Elliott et al. 2019, and it's showing values of delta 15N for NOx species on the left and the delta 15N for ammonia species on the right by different emission sources. And you can see that the NOx source values tend to be more positive. So here's the range of delta 15N values at the bottom of each figure. And so these emission sources tend to be more positive than the ammonia deposition sources. However, there's quite a bit of variation for each of these sources, as well as overlap between the sources, which makes things quite tricky. So as I mentioned earlier, lichens are totally reliant on atmospheric sources of nutrition. Not only can lichen nitrogen tissue content be correlated with ambient levels of atmospheric nitrogen, but lichen delta 15N can be used as an integrated measure of the delta 15N of atmospheric deposition. So this concept comes from foundational work done over in Europe in ambitrophic bogs, which are peat accumulating ecosystems that are fed only by atmospheric deposition. This figure here is from Bragaza et al. 2005, and it's showing the delta 15N of sphagnum peat moss along the y-axis, and along the x-axis is the NHX to NOx ratio in atmospheric deposition, which is the ratio of reduced to oxidized nitrogen compounds in both dry and wet deposition. So sphagnum plants had delta 15N signatures ranging from about -8 to -3 per mil. And they found that isotopic signatures were related to the ratio of reduced to oxidized nitrogen forms in atmospheric deposition rather than to the total amount of atmospheric deposition, which indicates that delta 15N signature of sphagnum moss can be used as an integrated measure of delta 15N of the atmospheric precipitation. They found that bogs located in areas characterized by greater emissions of ammonia, which if you remember from the beginning of this talk, are mainly affected by agricultural activities, had sphagnum moss with lower delta 15N compared to bogs located in areas dominated by nitrogen oxide emissions, which are mainly derived from industrial activities. So these findings suggest the type of nitrogen prevailing at a given locality as a consequence of human activities significantly influences the delta 15N signature of the mosses. So looking at my data collected from the North Cascades, focusing again just on one of the collected lichen species, Platysmacia glauca, we see that delta 15N was very negative, ranging from -6.9 to -4.1 per mil. And the lowest values are dark blue, and then red indicates the most positive values. We see that delta 15N tended to be more positive with increasing elevation. We also see certain areas of the park that have more positive values, including the Cascade Pass area that I highlighted earlier. If you recall from that land cover map I showed earlier, this region of the park is in the closest proximity to the Seattle metropolitan area. So with more positive delta 15 nitrogen signatures, this suggests that oxidized forms of nitrogen derived from industry and vehicle exhaust may be a significant component of atmospheric deposition in this area. And for other areas of the park, with more negative delta 15N signatures, this suggests that reduced forms of nitrogen derived from agricultural sources may be a larger component of nitrogen deposition in these regions. So the next step for this project is to integrate lichen data and atmospheric chemistry models in a GIS framework to develop a powerful approach to evaluate nitrogen critical load exceedance in the Pacific Northwest. I will not only be incorporating the lichen data that I collected from the North Cascades, but will also be incorporating data from U.S. Forest Service air quality plots in Washington and Oregon. Once we have developed a methodology, we'll hopefully be able to assess patterns of nitrogen deposition across the Pacific Northwest region and assess the factors that are influencing those observed patterns. As I mentioned earlier, previous analyses have shown that modeled nitrogen deposition has a high degree of uncertainty in mountainous regions. And with this approach, we expect greater accuracy of nitrogen deposition estimates in these regions due to the greater spatial distribution and density of air quality sampling with the integration of lichen data. Regional lichen nitrogen stable isotope composition will help us gain insights into emission chemistries and predominant sources of nitrogen emissions impacting the region. And ultimately, the goal is to have this study be a template for assessing nitrogen deposition and nitrogen critical loads across mountainous regions throughout the US. So with that, I would like to acknowledge my funding sources and collaborators, as well as my amazing field crews that I had in the North Cascades for 2018 and 2019, including Heather Stewart-Ahn, who will be giving a talk later today. And with that, I'll take any questions. Thank you.
Wonderful job, Meaghan. That was fantastic. I've got quite a few questions for you here. First, from Lolita, we have, how does N fixation impact N isotope ratios? Have you found that the presence and abundance of N fixing lichen influence N isotope ratios?
Yeah, that's a great question. So right now with my project, I'm really delving into all the different things that may be influencing the delta 15N of the lichens I collected. So you have obviously the variation in sources, which I mentioned, of the actual emission sources. Then you have the transformations that occur in the atmosphere. But then once deposition occurs on the landscape, you have canopy effects, and that could also include the prevalence of cyanolichens that fix atmospheric nitrogen in the canopy. And that is going to be an additional nitrogen source that may, reach the lichens that I collected. And that does have a different isotopic signature than the different emission sources that I'm looking at. It actually, I think if you saw on one of my slides down here, I kind of have this very coarse range of different lichen delta 15N values where nitrogen fixation is right around 0 per mil. Then fossil fuel emissions tend to be more positive and agriculture tends to be more negative. And so this nitrogen fixation from cyanolichens, it's going to be part of the mix that I'm going to need to account for. But I haven't done those analyses really yet, but it'll be interesting to see. As Heather can attest to, there's definitely different plots where we sampled where there is way more biomass of cyanolichens, so to see how those plots differ from other areas where cyanolichens might be absent, such as in higher elevation areas.
Thanks. We have another question from Diane. She's curious if you're looking at any other elements.
So no, unlike Adrian, who was talking about the ICP analysis that she was doing in her lichens, I'm just focusing on nitrogen. I do have data for carbon as well that I could look at, both carbon content as well as the carbon nitrogen stable isotope composition. But other than that, at this point, I'm not looking at any other elements.
I have a question. I have a question from John. Mm-hmm. I believe this was actually addressed by your colleague, Elise. Can you do stable isotope analysis on historic herbarium samples?
Ooh, yeah. I hope that Elise connects with him to talk about that because that's exactly what she did for a project along with me in our lab where she collected a bunch of Loberia oregana herbarium specimens and was doing the stable isotope composition on them. There are some considerations that you have to account for with that. One of which is those specimens weren't originally collected with the intention of doing any kind of elemental analysis on them. So they didn't have the same kind of sterile collection techniques that we use with the US Forest Service protocol that a lot of you are familiar with. So wearing gloves, avoiding any contamination of that kind. But yeah, there have been several studies that have been able to look at these archival samples and look at potentially deposition over time, as Elise was doing. So yeah, I think it's a really fascinating area of research. And, as long as you can maybe account for some of those things, it could be very useful for, yeah, interpreting data.
I believe we have time for two more questions. And Bruce asks, do you see critical loads as arbitrary cutoffs or as true thresholds where a small change in inputs makes a big difference in response?
Yeah, that's a really great question. I do think that, nitrogen critical loads are being used where they can be very useful for resource managers to determine where these detrimental effects might occur. So lichen-based nitrogen critical loads have been evaluated based on a number of things, such as species richness or the change in lichen species composition, or even the change of different functional groups, such as cyanolichens or matrix or forage lichens, so lichens that have different ecological roles. And so, it maybe is a little bit arbitrary, the fact that you can, have a different critical load based on what you're looking at, what component of the ecosystem are you considering that, most important thing that a harmful effect to that is going to determine your nitrogen critical load. But I don't think that takes away from the utility of them. But yeah, they are more of a gradient than maybe this like very strict threshold.
I'm going to go ahead and ask one more question. Apologies to everyone else. Jess wants to know, how long of a time period do estimates of nitrogen deposition use lichen nitrogen content integrate over? For example, do they represent nitrogen deposition over 1 year, 5 years, etc.?
Yeah, we definitely don't know the exact answer to that question, but using the nitrogen content of lichen tissue, we are assuming that it's integrating over multiple years. Exactly how many years, we're not sure, but probably something around five years. But of course, it's going to depend on the age of the lichen tissue. That's why for the Forest Service protocol, we're collecting a fairly large biomass of lichen tissue that we then homogenize, and that's going to represent a bunch of different age classes so that sample could be representative of kind of like the average of a plot rather than, just taking very young lichen tissue that might have only captured nitrogen deposition from the past year.